Hi and welcome to our first episode. So today we're going to be talking about biological molecules and carbohydrates. Before we start, I just want you to be aware that this video is going to cover things in general. It's not going to cover everything you need to know, so make sure you check your exam board specification to know that you've covered everything. It looks a bit of a mess and a bit horrible, but honestly, once we go through it, hopefully at the end you'll think it's okay. So let's get started then. So why are they making us learn this firstly? Well, it's to do with the fact that all of these biological molecules are common between species. Okay, you find carbohydrates in both people and plants. You find proteins in all species as well. So when we think about our molecules, most of them exist as polymers. These are formed from monomers. So our monomer is our single unit. When they join together, will become our polymer. So examples of monomers that we need to know are the monosaccharides, the nucleotides, and the amino acids. Now somehow they form our polymers, and they do this through a condensation reaction, which we'll talk about in a second. We also need to be able to break down our polymers, and a prime example of that is when we digest our food. So let's start off with the first reaction we're going to look at, which is how we're going to get a monomer to form a polymer, and this is our condensation reaction. So let's think about this reaction in particular. What is really going on? So as you can see, we've actually got two monomers next to each other. Then they form a bond, and we'll talk more about how this bond varies depending on which monomers we're using. But what we really need to understand is the fact that we can have this OH group on this one molecule and this other OH group on the other monomer. And essentially, the condensation reaction involves the removal of water, and we get this bond here. I think the easiest way to remember a condensation reaction is that it produces water. A bit like how when you learnt in GCSE, the condensation reactions, an example would be, when hot air hits cold glass or something on a window, and then you get water forming out as a result. You get that droplet of water. That's condensation too. But what about the reverse? How do we break things up? Like we said, we use hydrolysis reactions. And this is this reaction going on right here. Now, I've put them next to each other just to show they are basically the reverse. And instead of water leaving, we're putting water, or in fact, we're using water to break a bond. That is what hydrolysis is. And as you can see, we're getting that bond there with the oxygen. We're adding the water, and the H2O will then create those two OHs at the ends of the new monomers we've got here. It's important to remember that, we're talking about monomers, but the thing is, the reason I've left these ends empty here is that we can keep doing this reaction again and again and again and again with adjacent monomers, and that is how we get our really big polymers. So just to summarise the biological molecules bit of the monomers and polymers, many monomers can join together to form a polymer. They do this through a condensation reaction. In contrast, when we want to break down our polymers, we use a hydrolysis reaction to then get our monomers. So let's get started at looking at our next topic, which is going to be on carbohydrates. Put simply, carbohydrates are basically the sugars of the food world, but they are a great energy resource, mainly supplying glucose when it comes to the whole respiration thing that you need to learn, which I'll come to a bit later. Now remember how we were talking about how monomers join together to form a polymer? Well, when it comes to carbohydrates, they've got specific names. So when we're thinking about a monomer, when it comes to the sugar, we use a monosaccharide as the term for it. So this monosaccharide can vary. Prime examples we need to be aware of are fructose, galactose, alpha glucose, and beta glucose. This is alpha glucose and beta glucose. Right, I know some of you might be thinking, okay, look, I'm doing biology, not chemistry. Why do I need to know the structure? Well, it's actually important for its biological function, which we'll get onto in a second. But just by looking at it, you might think, wait, aren't these exactly the same? No, okay, they are almost exactly the same, apart from the fact that this part, the OH and the H, if I got a better pen maybe, that's better, are different and basically flipped over in a sense. Now, if you do chemistry, you know this is important because there's not double bond, we don't have any rotation. So these are actually isomers of each other. Another thing that we didn't mention before is that we don't just get from the mono part to the poly part. Whenever we join two monosaccharides together, we get a disaccharide. Di meaning two. There are three that we need to be aware of and what monomers they're formed from, or monosaccharides, I should say. Maltose, sucrose, lactose. So with maltose, we get that from two glucose molecules. From sucrose, we need glucose and fructose, and for lactose, we need glucose and galactose. If in doubt, put glucose, and it might be something else. But when we keep adding more of those molecules together, so if we add more disaccharides, or even add another monomer to the disaccharide, we get a polysaccharide. These are the three we need to know here. Starch, which consists of amylo and amylopectin, glycogen, and cellulose. Now, you can tell from the fact that I've drawn a lot of things down here, we're now going to go into some more detail. But that was a brief overview. So let's start off with the monosaccharides, because they should be the most straightforward. And as well as just knowing what alpha glucose is, beta glucose, fructose, and galactose are, if we then look at this condensation reaction, this is where we're going next. We are getting the polymer from the monomer, or firstly, we're getting the disaccharide. So, remember how we were talking about the OHs? Well, let's consider if we join them together. This is the alpha glucose and the beta glucose joining together, which in more detail than you need to know, it is a form of maltose. But as you can see, what's really important is I've outlined the fact that water is being taken out. This is our condensation reaction. Okay, what's left? That oxygen, and that oxygen is going to form a bond or a bridge between the two. And that bond, when it comes to carbohydrates, is known as a glycosidic bond. This will happen between those molecules, the glucose and fructose for sucrose, the glucose and galactose for lactose, and that's how we get our disaccharides. Okay, so the last thing that we're going to do is just talk about the polysaccharides. Let's start off with starch. I've drawn a flower here, not the prettiest, but it's there to symbolise the fact that we find starch in plants. Okay, they consist of being either an amylose or an amylopectin. Now, starch is made from alpha glucose, okay? And the amylose itself is this spiral shape, which makes it really good for storage as well. Another important thing about starch, both amylose and amylopectin, is that they are insoluble. This is really helpful because if they were soluble, it would affect the water potential of our cells in the plant, which could draw water in and out certain places, and essentially it wouldn't be able to survive and probably die. Now, amylose, the reason it's coiled is because its glycosidic bonds are 1,1,4. Amylopectin, in comparison, has these branches. Now, these branches occur at glycosidic bonds 1,6. This is quite a bit of detail, but I think it's really important to understand why they're different. So the different bonding leads to different structure. That's important for both biology and chemistry. And here, the branching actually makes it more accessible for enzymes to work on. So just to clarify, we actually do have 1,4 bonds in our amylopectin, but where we get the branching is where we have our 1,6 bonds. And what's interesting about starch? Do you remember that potato thing that you did with the iodine test? And it made everything go blue-black when there was starch? Well, that's one experiment that you need to be aware of and need to know when it comes to exams and identifying starch. So hang on a minute, what about glycogen? Yes, that is also another form of alpha glucose, but can you see that in comparison to our amylose and amylopectin, that is much more branched. It's got more of those 1,6 bonds. And that is really important because I said before, it makes it more accessible for our enzymes to break down and get glucose as monosaccharides, which can then be used for respiration. So if we've got more branching, we're increasing the surface area, the enzymes can work more efficiently, and that means we get a greater amount of glucose per unit time with this branching. And just to show, that it's human, I've drawn a lovely artistic face. Anyway, Now what about our last polysaccharide? Cellulose. Already, cellulose you should think cell walls. Now this is different from starch and glycogen because it uses beta glucose, not alpha glucose. So, beta glucose here, why is that helpful? Well actually, it ends up forming, when in polysaccharide form, the beta glucose molecules actually form this long straight chain. And these chains kind of pack together. And when they pack together, we get hydrogen bonds forming between. Now these hydrogen bonds by themselves are weak, but together they're quite strong. And as you can see, if we've got lots and lots of bonds, signified by my lovely red, I don't even know what they are, dashes, this ends up joining all of those straight molecules together and forming microfibrils, which will give our cell wall its strength. The last thing that we need to know when it comes to carbohydrates are the experiments for reducing and non-reducing sugars. So it's actually not important to really know the difference between reducing and non-reducing, more so the experiments that we can do to prove it. If you look it up yourself, it might help you, something to do with ketones and some more interesting things. We'll look at practicals in more detail later, but just to get a basic overview of what this experiment is about, what we're doing is we're adding Benedict solution, yes, like Benedict Cumberbatch, but not like Benedict Cumberbatch, and heating the solution. So if we have reducing sugars present, after you complete that Benedict's test and, add the Benedict solution and heat it up, you end up with a brick red precipitate. Technically there's kind of a spectrum, so it doesn't just go blue or brick red, you kind of have this yellow in between or a greenish colour. And that's really important to understand, that it might give an indication of how much sugar's there. It also could mean, you haven't heated it up properly or carried out the experiment fully. But the gold standard is that we have a brick red precipitate formed. So if that doesn't work, which will be quite sad, you just end up with blue, you take another sample of it, so a different sample, add some hydrochloric acid, add some sodium hydrogen carbonate, and then do the test again. You then add the Benedicts and then heat it up. And if you then get your brick red precipitate here, you've got a non-reducing sugar. And if you don't, then unfortunately, if you tested negative for both the first test and the second one, you don't have any reducing or non-reducing sugars there, which is quite sad, as you can see. But this second test, hydrochloric acid, sodium hydrogen carbonate, that's a neutralization reaction going on there. Now precipitate can kind of block light, so we can actually indirectly measure how much precipitate we've got through using colorimetry. But again, more on that later. So that was just an overview of the biological molecules and the carbohydrates topics. I hope that helped. And I never thought I'd say this, if you liked that video, make sure that you like it by clicking that button and subscribe. Thanks guys.